previous lecture we have started with the methods based upon excitation, that is detection and measurement of radioactivity on the based upon the methods on excitation. We have started with scintillation counting, where we have discussed about solid scintillation counting. In solid scintillation counting, you have to keep the sample very close to the solid crystal floor, and there is a photomultiplier tube put in close to the crystals. So, solid scintillation counters are mostly utilized for strong beta emitters or usually for the radioisotopes with gamma rays emission, as it is quite widely utilized in lot of different biotechnological applications. In this lecture, we are going to start with another scintillation counting method, which is the most widely used, that is liquid scintillation counting method. Now, GM counters and solid scintillation counters are inefficient for low energy particles, particularly for 3H, which is widely used in different biotechnological applications. Now, two major factors limiting their efficiency are that not all emitted particles reach the detector and not all are counted from those that reach the detector. Now, main reason for not reaching the detector is that the geometry is such that some particles are emitted in a direction that misses the detector. For example, in a sample placed before GM tube, one half of the particles will be emitted in a direction away from the detector, because only 50% will really enter the tube, 50% will go in another direction. So, geometry can be a problem or could be a limiting factor. Now, reason for not detecting low energy particles which reach detector is that the particle have insufficient energy to cause ionization of the gas. So, when they reach there is they do not carry sufficient energy to ionize the gas. So, that could be the another reason. The problem could be solved and efficiency of low energy particles could be increased substantially if samples are contained within the detector and problem of self absorption can be avoided. So, a solution is provided by the technique of liquid scintillation counting. So, what we are here can be done that those radiations which are lost or those 3H particles which are lost could be avoided by putting the sample within the detector. Now, liquid scintillation counting is a very standard laboratory method, widely used method like I said, in which the sample is mixed with a scintillation cocktail containing a solvent and one or more floors. We will be discussing this in detail. So, what is done is that whichever radioactive samples you are going to count, they will be put in this cocktail, that is they could be dissolved in this scintillation cocktail or they could be suspended in this particular solution. Now, this cocktail contains an aromatic solvent which is historically benzene or toluene has been used, but more recently less hazardous solvents have come into favor. And apart from this solvent, they will be containing very small amounts of other additives known as fluors. Now, beta particles emitted from the sample will transfer their energy to the solvent molecule, which in turn will transfer their energy to the fluor. So, what is happening? First is very weak beta particles are transferring their energy to the solvent, and then that particular energy is being given to the fluors. We will see how this is done. Now, the excited fluor molecules will dissipate the energy by emitting light, and this light will be collected by photomultiplier. Now, the whole thing is explained quite well by this diagram here. If you see here, there is a radioactive material here, which is sample containing radioactive material or radioisotope. Now, here it is seen that a beta particle is emitted and this beta particle will excite the solvent molecule. Now, this excited solvent molecule on de-excitation will emit photons or light and they will be taken up by the floor, which is it could be one floor or it could be two floors. If there is two floors, then one is called primary floor and another is called secondary floor. So, if there are two floors, the light will be gone to primary floor and then from primary floor to secondary floor, and then the light emitted by these floors either primary or secondary will be taken up by the photomultiplier tube and finally analyzed. So, this is how the whole process in liquid scintillation counting occurs actually, as the radioactive sample is present in the scintillation cocktail only. And as I have shown you earlier in previous lecture, that how the sample is placed very close to the detector. All right. This is the photomultiplier tube. This we have explained earlier in case where we were discussing scanning electron microscope also. So, there is a scintillator which collects this light, these are converted to, and through light guide they reaches to the tube. There is a photo cathode. These photons are converted to photoelectrons, they are amplified and finally signal is measured. All right. So, what we are looking at is that how this energy transfer is taking place in liquid scintillation counting. Now, a small number of organic solvents fluoresce when bombarded with radioactivity, like we have seen in the previous figure. Now, the light emitted from this solvent is of very short wavelength. So, this will not be efficiently detected by photomultiplier. I will show you this in a little while. However, if a compound, that is a fluor at low concentration, is dissolved that can accept the energy from the solvent and itself fluoresce at a longer wavelength, then the light can be more efficiently detected. Now, such a compound is known as primary fluor, and the most frequently used example is PPO or 2,5-diphenyl oxazole. This is a very common primary fluor. Now, so what happens? The light has first a solvent is excited, then it emits longer wavelength which is taken up by primary fluor, it is excited, then again it will emit further longer wavelength. Now, it depends on the sensitivity of the photomultiplier tube whether it can accept directly that wavelength or not. Now, if wavelength of the photon emitted by the fluor, that is primary fluor, are not in the region of highest sensitivity of the tube, then the electric pulse will not be generated. So, what you require? You require a secondary fluor in the vicinity that will absorb the photon emitted by primary fluor and then will re-emit them as a fluorescence at a longer wavelength, which could be detected efficiently by photomultiplier. So, little bit let us understand this on your screen. What is this sensitivity we are talking about, and if I could little bit tell you about this. So, what we are talking about is that the whole process of energy transfer has to be detected actually, and like I said, beta emitters which excites the solvents, the wavelength is not long enough to be in the region of photomultiplier tube sensitivity. So, what you have is you have to have that particular emitted radiation in the sensitive region of the tube. So, all right. So, if we say this is the sensitive region of the tube, that is the sensitive region or sensitivity of the tube, that is the sensitivity of tube, that is photomultiplier tube. Now, let us consider the energy transfer. So, what happens? Beta particle which is emitted from say 3H will excite the solvent, all right. So, solvent is excited here, which is could be whichever solvent is present in here, and if we just have to give certain wavelength, say it is at around 250 nanometer. Now, if you can see this particular one is not really in the sensitivity zone of the tube. Now, this solvent will excite the primary fluor, and the primary fluor is excited in here again, and you can see this primary fluor, though it is in somewhat in the region, but it is not it will not be efficiently collected or this wavelength will not be efficiently collected by photomultiplier and electric signal may not be generated. So, this is primary fluor, or which we were talking about, that is PPO. Now, so if the light could not be efficiently collected from the primary fluor, then it will not be able to generate and efficient measurements cannot be taken place. So, what you require? You require a second fluor, which will be called secondary fluor. So, then there will be a secondary fluor, which is, you can see here, this is in very much in the that particular region, that is sensitivity of the tube, and if this is this particular secondary fluor could be one which can efficiently transfer this light or the particular wavelength which is in this region, longer wavelength, that could be taken up or which could be read by the photomultiplier tube. So, this is what we are talking about in terms of the energy transfer, or that is efficient energy transfer right from the 3H radioactive material till the secondary fluor and to photomultiplier tube. All right. So, let us return to our discussion. So, what we are discussing was that you can require 2 fluors rather than 1, but it could be that 1 fluor could also work, like in many advanced versions it has this particular facility. Now, so unfortunately, the light emitted by PPO, like we were talking, is not always detected. So, you have a secondary fluor, and like I said, secondary fluor is POPOP, which is 1,4-di-2,5 phenyl oxazole benzene, and we will call it POPOP. Now, both fluors act here, they are simply acting as wavelength shifters actually, and they do not absorb original energy of the beta particle. So, use of primary fluor is mandatory, because secondary fluor cannot absorb the energy directly from the solvent. So, why not use directly secondary fluor, which is in the sensitive region of photomultiplier tube, because this will not be able to take up the wavelength which is emitted by the solvent directly. So, therefore, you require a primary fluor also. Now, secondary fluors may be unnecessary with modern advanced scintillation counters, where for example, compounds such as butyl PBD could be utilized and you do not require a secondary fluor. So, essentially what you have is, if we summarize the whole thing, that is the energy transfer process, then it becomes something like shown in here. So, you have radiation in terms of beta particles, say from a very weak radioactive material, that is radioisotope 3H. Now, this radiation is taken here, there is a solvent, this is taken by the solvent, it is excited. On de-excitation, it will release some light or particular longer wavelength, which will excite primary fluor, which is excited. Again on de-excitation, it will emit certain longer wavelengths, which will excite the secondary fluor, and then on de-excitation again it will emit further longer wavelength, which will be read by the photomultiplier tube. So, that is the whole summary of energy transfer in here. Now, the efficiency of detection of original radioactive decay will depend on many factors, and some of these factors will be, one is the properties of solvent, in that the fraction of the absorbed energy leads to the excitation of solvent molecules. So, properties of solvent would be very important, and then the part of the excitation energy is transferred to the primary fluor rather than being dissipated as heat. So, one very important part is what are the properties of solvent, that in that it can be excited suitably, efficiently, and also then it can transfer the energy to the primary floor without being wasting that particular energy being wasted as heat. Second is the number of photons produced by primary and secondary floors, like how efficiently that energy transfer takes place. Then efficiency of the photomultiplier tube to gather photons, that is also very important, and that will depend on design or geometry of the tube. So, the design of the photomultiplier tube is also important. Then signal to noise ratio of the photomultiplier is another important factor. Finally, efficiency to convert the charge of photomultiplier into the electric pulse will also determine the efficiency of detection. So, these are some factors which will be determining the efficiency of detection of original radioactive event. Now, scintillation counters have lot of advantages over GM counters, which we have discussed earlier. Now, those advantages, if we count over gas ionization, one is much higher count rates are possible in scintillation counting as compared to GM counting, because of the rapidity of fluorescence decay, which is almost in the order of 10 raised to power minus 9 seconds. And when you compare it to the dead time in the GM tube, which is 10 raised to power minus 4 seconds. So, the rapidity of or much higher count rates are possible because of the rapidity of fluorescence decay. Then much higher counting efficiencies are achieved, particularly for low energy beta emitters. Now, the best efficiency for 3H would be 5% with GM counters, and compare it with the scintillation counters like liquid scintillation counters, it is about or more than 50% efficiency could be achieved routinely. So, you can compare that the it is much efficient than the GM counter. Then ability to accommodate samples of any type, like including liquids, solids, suspensions and gels, that is another important part of liquid scintillation counting. Ease of sample preparation, the ability to count separately different isotopes in the same sample, that is a big advantage in terms of if certain sample is containing two isotopes, that could be easily distinguished if the energy spectrum is differing or different for two isotopes. So, that could not be done in GM counters. Then scintillation counters are highly automated now and are very good for quantitative and qualitative estimations. So, there are a lot of advantages of the scintillation counting, particularly liquid scintillation counting, but there are a lot of disadvantages also. Now, for scintillation counting, most of the inherent disadvantages have been overcome by improvement in instrument design, but some of the disadvantages, which includes one is cost per sample of scintillation counting, that is very high, but like I said, this factor could be overlooked as it provides versatility, sensitivity, accuracy and ease for most applications. So, it could be overlooked, the cost of the operation could be overlooked because of lot of different advantages in here. Now, there are other important disadvantages. One is photomultiplier noise, actually that is at very high voltage applied to the photomultiplier, the electronic events occur in the system and these are independent of radioactivity, but they will contribute to a high background count referred to as photomultiplier noise. So, that is photomultiplier noise is one problem. Now, this photomultiplier noise could be partially reduced by cooling the photomultiplier. Apart from cooling, which may not be enough for reducing the noise, there could be, you can put in a pulse height analyzer. Now, pulse height analyzer is set to reject most of the noise pulse which are of low energy, that is a threshold or gate setting is done. Now, this disadvantage could be that low energy pulse resulting from low energy radioactivity like 3H could be discarded. So, what is done is there is another method is used which is called coincidence counting. what is done here is two PMT tubes or two photomultiplier tubes are set in coincidence such that only when a pulse is generated in both tubes at the same time it is allowed to pass to scalar, and this will certainly reduce the noise to a very low level. Now, a coincidence circuit is set to discriminate between noise and pulse caused by beta particles. Only in the later case will light strike both detectors simultaneously, and the coincidence gate will allow or announce that a beta decay has occurred. So, if it is a noise, then possibility is that it will not hit both tubes. Now, the chance of noise pulses reaching both photomultiplier tubes simultaneously is very rare or very small. If the coincidence circuit detects a pulse from PMT or photomultiplier tube and not from the other within 40 nanoseconds, the analyzer will disregard the pulse. So, this coincidence circuit ensures that the background, that is liquid scintillation counting background, is kept at as low as possible here. This figure shows how two photomultiplier tubes have been put in here. You can see there is one photomultiplier tube here, another here, and the sample is in between. So, when signal reaches to both, then only it will be counted, otherwise it will not be counted. So, that is the one method to reduce the photomultiplier noise. Now, there are other disadvantages also. One of the greatest disadvantages of scintillation counting is quenching. Now, this occurs when the energy transfer process suffers interference. So, quenching refers to any process which decreases the fluorescence intensity of a given substance. So, in essence it is interfering with the quenching, the phenomenon of quenching interfering with the energy transfer actually, and it will certainly lower the counts. Now, quenching can be of different kinds, and these kinds could be, one is optical quenching. Now, optical quenching occurs when dirty scintillation vials are used. What will happen? If these are dirty scintillation vials are used, they will absorb some of the light being emitted before it reaches the photomultiplier. So, what has to be done? That one needs to use clean vial, and they should be handled carefully to avoid any grease or dirt on the vial surface. So, optical quenching is one problem. Then there could be others like color quenching. The color quenching occurs if the sample is colored and results in light emitted being absorbed. So, if color is absorbing the emitted light, then again that is a problem with the scintillation counting. And so, what has to be done is that if in some way if color could be reduced or certain like relative counting could be done, it could be then taken care of. So, color quenching is another problem. Now, third quenching problem could be chemical quenching. Now, this form of quenching occurs when anything in the sample interferes with the transfer of energy from the solvent to the primary floor or from the primary floor to the secondary floor. It is the most difficult form of quenching to accommodate. Now, an appropriate method of standardization which requires determining the counting efficiency of each sample and then conversion of counts per minute to absolute counts has to be done, which is kind of adds to your problems. There are other things like chemiluminescence. Now, chemiluminescence results from chemical reactions between components of the sample to be counted, and the scintillation cocktail and produces light emission unrelated to excitation. So, of the solvent and flow system by radioactivity. Now, these light emissions are generally low energy events and they are rejected by the threshold setting of the photomultiplier in the same way as a photomultiplier noise. So, chemiluminescence could be another problem where certain chemical reactions are occurring and they are producing light which is totally unrelated to excitation, and it could be, if it is a weak emissions, then they could be easily taken care of by a threshold setting actually. Like chemiluminescence, there is another phenomenon called phospholuminescence. Now, this results from the components of the sample including the vial itself absorbing light and re-emitting it. Now, unlike chemiluminescence, which is once only effect, phospholuminescence will occur on each exposure of a sample to light, and samples that are pigmented are most likely to give phosphorescence. Now, to sort out this problem, samples should be dark adapted prior to counting, and sample holders should be kept closed throughout the counting process. Now, despite all the complications described above, which are quenching and other problems, scintillation counters are universal in biosciences or biological sciences departments. They are widely used and they are very useful for different applications and for counting, particularly counting the weak emitters. Now, like we said, in one of the advantages was that you can count 2 samples at a time, that is 2 isotopes at a time. So, scintillation counting can be used for dual labeled samples. A great advantage of scintillation counting is ability to determine the ratio of 2 or more radioisotopes present in a mixture. Now, this is possible because the size of the electric pulse produced by the conversion of light energy in the photomultiplier is related directly to the energy of the original radioactive event. The resolution of 2 isotopes is made possible with a pulse height analyzer. Now, what is the pulse height analyzer? It is an electronic equipment that can sort pulses of fluctuation in current or voltage in terms of its magnitude. Now, this is equipped with discriminator that controls. So, discriminator controls or that determine the voltage levels, defining the voltage range actually, and most of the time 2 discriminators are usually employed. Different beta emitting isotopes have different energy spectra. It is possible to detect and quantify 2 isotopes separately in a single chamber, provided their energy spectra are sufficiently different. Now, if we take some examples of pair of isotopes that have sufficiently different energy spectra, they could be like 3H and 14C, 3H and 35S, 3H and 32P, 35S and 32P. If you could recall, I have shown you in previous lectures the beta spectra or beta particle emission spectra of different isotopes, and they were quite different in all the cases. Now, like I will show you one figure here, but before that, now this spectra of 2 isotopes which is could be there, they will overlap only slightly. What does that mean? That they are sufficiently different. Now, by setting a pulse height analyzer to reject all pulses of an energy below certain threshold, like say threshold x, and reject all pulses of energy above certain threshold, say y or window y, you will be able to set a particular threshold value, and also you will have for another isotope, you will have a threshold of another threshold, say threshold A, and another window, say window B. Now, it will be possible to separate these 2 isotopes completely. So, what a pulse height analyzer set with a threshold and window for a particular isotope, this is known as channel actually. So, what you have done, because you would like to detect and quantify 2 of the isotopes, you have certain threshold values with a pulse height analyzer set with threshold values for 2 isotopes, and this particular thing is called channel. Most modern counters operate with say they are multi-channel analyzers, and so they have maybe they can have more than 1 channels, and they are based on analog to digital converter. Now, dual label counting has proved to be useful in many aspects of molecular biology. Example, say it would be nucleic acid hybridization studies or mechanism of ribosomal subunits, protein synthesis, or transcription, metabolism, or drug analysis or development. A lot of different applications could be there. Now, this figure clearly shows what we were discussing here. So, what you have is a pulse you are trying to detect two isotopes. If you can see, there is X threshold and there is a Y threshold, which is Y window we call, and this part here that will be measuring this channel. One channel will be measuring isotope S and another channel 2, which is set on another threshold, which is A, and it will reject all below A, all signals below A, and there is a window B. So, there will be another channel for second isotope, which we are calling isotope T. So, by putting in two channels, that is multi-channel detectors, you would be able to detect and quantify the two isotopes simultaneously in a sample. So, that is a great advantage of liquid or scintillation counting over GM counting. Now, let us little bit move into the determination of counting efficiency. Now, a major problem encountered in scintillation counting is that of quenching. Now, this is a real problem, like we have seen or we have discussed it, and it makes it very important to determine the counting efficiency, because what you are doing is you have to standardize the counting methods, so that you know that every time you are counting correctly. Now, this could be done by one of the several methods of standardization, and all of which apply to both solid and liquid scintillation counting. Now, this could be internal standardization, there could be external standardization, and even there could be a channel ratios could be taken, like this could be counted in two channels and it could be ratios could be taken. Now, very briefly, in internal standardization, first the sample is counted and it gives a reading of, say, a particular count per minute. Then it will be removed from the counter and a small amount of standard material of known disintegration will be taken. Then that will be sample will be again counted, that will be C counts per minute, and then counting efficiency of the sample will be calculated by this formula here, which is 100 in bracket C minus A upon B %. So, you will be able to, through internal standardization, you will be able to count the efficiency here through internal standardization. Now, it is obviously necessary in this method to use an internal standard that contains the same isotope as the one being counted, and also to ensure that the standard itself does not act as a quenching agent. So, these are a few precautions has to be taken. Internal standardization is simple and reliable and corrects adequately for all types of quenching. Now, this method demands calibration of two factors and it demands more accurate pipetting in adding standards. So, these things has to be taken care of for internal standardization. Then there could be another way is channel ratios actually. Now, when a sample in scintillation counter is quenched, the scintillation process is less efficient. So, less light is produced for a given quantum energy of radiation. So, what happens then? The energy of spectrum for a quenched sample appears to be lower than for an unquenched sample. So, the higher the degree of quenching, the more pronounced is the resulting decrease in the spectrum. So, the method involves the preparation of a calibration curve based on counting in two channels that cover different but overlapping parts of the spectrum. Now, as a sample is quenched and the spectrum shifts to gradually lower apparent energy, the ratio of the counts in each sample will vary. To prepare the standard curve, a set of quenched standards is counted. So, the efficiency is then plotted against channel ratio to form the standard curve, and once the standard curve has been prepared, the efficiency of counting experimental samples can be determined, and samples are counted in the same two channels. The ratio is calculated, put into the graph, and the efficiency is read. Multi-channel scintillation counters operate on the same principle, but the whole shape and position of the spectrum is analyzed. So, these systems have greater precision than the two channel approach, as the whole of the spectrum is used for analysis. So, here it is shown in here where you can have counts per minute, and you can see how the different, due to different quenching effects, the spectrum or lower energy spectrum is obtained. So, this could be through channel ratio, it could be done. Another is external standardization. Now, in external standardization, instruments will have a gamma emitting external standard built into the counter. Now, under the control of the counter, each sample to be counted is exposed to the external source, which is automatically shifted from a lead shield to the counting chamber. Now, the gamma radiation penetrates the vial and excites the scintillation fluid. Then the resulting spectrum is unique to the source and is significantly different from that produced by the sample in the vial. So, the gamma source used varies according to the make of instruments. Now, the quenching agents present in the scintillation fluid will significantly affect the spectrum obtained. The instrument analyzes this spectrum and assigns a quench parameter to it. As for the channel ratio method, a standard curve is required, that is, range of quenched standard is counted, and then the external standard spectrum is analyzed in each case. Now, the resulting data are used to prepare a standard curve that is held in the instrument's computer, and unknown samples are then counted in the same way, and the efficiency is read from the standard curve and the sample counts corrected. Now, if you see the advantages and disadvantages, main advantage over channel ratio method being that it is suited to samples with low count rates. Now, disadvantage could be a standard curve is required for each set of circumstances, and user can be killed into a false sense of security. The system is so highly automated that it is easy to lose sight of the basic principle, and the method is not always appropriate. So, these are different methods which are used for standardization, that is internal standardization, external standardization and channel ratios. Now, so in this lecture what we have discussed here is the liquid scintillation counting. We have discussed about how energy transfer is taking place from radioisotope to solvent, from solvent to primary fluor, and from primary fluor to secondary fluor, and finally to photomultiplier tube. So, primary and secondary fluors in essence works as wavelength shifters in this case. Liquid scintillation counting or scintillation counting is a versatile method. It is widely used in different laboratories for radioactive measurements of weak emitters, and it is highly efficient method for both qualitative and quantitative estimations. Certainly, it needs standardization so that the efficiency of counting can be estimated, and that could be converted to disintegration per minute or per second, that is actual counting. In the next lecture, we are going to discuss about the sample preparation of liquid scintillation counting, and then we will be discussing about the third method, that is auto radiography for detection and measurement of radioactivity. Thank you.